Junior doctors, good evening. Shall we start with the recap? Doctors, I would like to check whether you did a revision of yesterday's block. We did for 30 minutes. Yeah, forgive me for being late because there is a technical glitch. Okay, so we'll start with the today's recap. What is the short-term goal to achieve our dreams? We are going to completely focus here for the next 30 minutes without any distraction. Okay, we are going to do the code words. So let us quickly start. Yesterday what we did in block 1, polyribosomes or polyribosomes, also called as polysomes. What is the meaning of polyribosome? There are several ribosomes attached to a single mRNA, and this chain is called as polysome. You have to remember this, attached to the single mRNA, not many mRNA chains. This can be a trap in the NEET exam. Next, what is the function of polysome? Helps in translation of mRNA into proteins. Where can you find the polyribosomes or polysomes? In the cytoplasm of the prokaryotic cell. Next, the second point, chromatophores. I told you to check the chromatophore yesterday, isn't it? It is an AR type of question. Okay, chromatophores. So what is the other name for cyanobacteria? Type it in the comments, students. I want the chat session to be an interactive one, but I am going to focus on the code words and I'll reply to you later, so you can type, you can ask me your doubts. Okay, even if you have doubt in the previous classes, or if you want me to take any chapter in the upcoming session, you can type it in the comments. I'll do it for you. Now, chromatophores, they are seen in cyanobacteria, also called as blue-green algae. It is a prokaryotic bacteria which can be mistaken for an eukaryotic plant. Okay. Next, what are chromatophores? They are the plasma membrane extension with the pigment. If it contains the pigment, it is called as chromatophore. What is the pigment? Chlorophyll A. So this chlorophyll A acts as the solar panel, and that is why cyanobacteria is able to synthesize its own food material, so we include that under photosynthetic autotrophs. Okay. Students, have you started? Did you have your pen and paper with you for today's block? Okay, today it's going to be a very interesting one. So main arena of cellular activities is cytoplasm, because all the metabolic activities takes place at the cytoplasm. You have all the important organelles embedded in the cytoplasm. Cytoplasm is very important for you to consider whether the cell is in a living state or in a dead state. Okay, cytoplasm. We saw one example, that is RBC. I hope you remember, RBCs are devoid of nucleus, but they have cytoplasm. What happens in today's question? I'll start with the question today. Okay, RBC. How do RBC get the energy? Okay, you have Krebs cycle, right? You have Krebs cycle for ATP generation. What type of cycle operates in RBC for the ATP generation? Today's question is this. Okay, type the answer quickly in the comments, students. I'll reply to your chat. I'm welcoming all the junior doctors here. Hello, who have joined. Hello from me to you, and happy to see your comment. So I'll reply to you once the chat is over, students. Next is nucleus. Nucleus is absent. If the nucleus is absent, it doesn't mean the cell is dead. Okay, so for a cell to be dead or alive, it depends on the cytoplasm. Okay, this one is the one which decides the living state of the cell. Next, under block 2, we saw about Golgi apparatus. What are the two phases of the Golgi apparatus? Doctors, please comment it. Please comment the answer. You have cis phase and trans phase. This is the entry phase, which is also called as the forming phase, which faces the endoplasmic reticulum. Next is the trans phase, also the transporting side, which is also called as the maturing phase, which faces the plasma membrane. Now I want you to type the answer for what is the function of Golgi apparatus. What is the function of Golgi apparatus? What is the function of Golgi apparatus? Next, under block 3, we saw about nucleus. Fleming is the one who named the material of the nucleus using the basics dye. He saw the nuclear material and he named it as chromatin. Next, perinuclear space. Before that, we will start from here. Okay, nucleus is covered by a 2-layered membrane. You call it as nuclear envelope. Envelope, like how envelope, you can see the letter, right? Letter, a covering letter. A cover for the letter. A cover for the letter. That's nuclear envelope. It is made up of 2 membranes, that is the outer membrane and inner membrane. In between the 2 membranes, you have a space, you call it as the perinuclear space. Peri means around. So the space is present continuously around the nucleus. You have to understand that. What is the numerical value of the space? 10 to 50 nanometer, nm. This one is important for NEET exam. Next, the outer membrane is continuous with which structure, students? Type it in the comments. It is continuous with the endoplasmic reticulum, which has ribosomes attached to it, which means it is a rough endoplasmic reticulum. Okay. Now, what is the function of this nucleus? Okay. You have cytoplasm, cellular cytoplasm. You have nucleus here. So, to prevent mixing up of this, okay, the nuclear envelope acts as a barrier. So now the question comes, okay, how does nucleus communicate with the cytoplasm and with the other organelles? What is the way it communicates? Type the answer in the comments, students. And today we are going to do 3 blocks, very interesting blocks. Okay, what are lysosomes? Name the organelle which is also called a suicidal bag. Name the organelle which is called a suicidal bag. Type the answer in the comments, doctors, my junior doctors, my team doctors. Comment the answer in the chat session. Even if you watch a replay, you can leave your answer in the comment section. Today the block 1 is lysosomes. Okay, first we are going to see about lysosomes. Second, about the plasma membrane, that is the composition of the plasma membrane. Then block 3, I am going to tell you the heading and you have to type the answer now. What are the exceptions to cell theory? What are the exceptions to cell theory? If you know the answer, type it in the comments, students. So first you have lysosomes. This is a vesicular structure. If they ask you, see the word vesicular. Vesicular means like this, surrounded by a membrane. Inside that, these lysosomes have enzymes. Okay, they call it as what? Hydrolytic enzymes. What is the meaning of hydrolysis? Now I want you to explain hydrolysis. What is the meaning of hydrolysis, doctors? If you know the answer, type it in the comments. Type it in the comments. What is the meaning of hydrolysis? Are you all just by hearting hydrolytic enzymes, hydrolysis? Okay, are you doing that? Now I will see the explanation. Now you will understand. Okay, this is a chemical process where a bigger molecule is converted to a smaller molecule. Okay, a chemical reaction in which a bigger molecule is converted to a smaller molecule. This you know, right? So by the addition of a water molecule. This is what hydro means water, lysis means H2O. Lysis means split. Okay, so by addition of your water molecule, you are breaking down a bigger molecule into a bigger substance into a smaller one. This is called as hydrolysis. The enzymes which speeds up this reaction are called as hydrolases. The whole group is called as hydrolases. Hydrolytic enzyme means you are calling an individual enzyme. Okay, hydrolytic enzyme. So basically what type of enzymes are this? Breaking down by adding a water molecule, by addition of a water molecule. I hope now you won't by heart, you will understand. So hydrolases, hydrolytic enzymes. So lysosomes, if you see the code words for lysosomes are hydrolases, hydrolytic enzymes and acidic pH. Now I wish you have to tell me, like what is the acidic, neutral, acidic, neutral, acidic, neutral and alkaline ranges. Alkaline ranges. Can you tell me the ranges, students? Neutral is 7. Okay. 7 to 14 is alkaline. 0 to 6.9 is acidic. So, because why I am mentioning here, it is a trap. There is a trap here. You know, you'll be studying acidic pH, but if they give you the value, I mean the enzyme like lipase or active at the pH of 5, you should know the value. If you don't know whether the 5 is acidic or alkaline or neutral, you cannot choose the right answer. So I have decoded this part also, 0 to 6.9. So this acidic pH means it's close to 5. The hydrolytic enzymes are close to 5, the pH range of 5. Okay, so hydrolases, hydrolytic enzymes, acidic pH means you have to choose lysosomes. Shall we write it 3 times, students? Follow with me. Hydrolases, hydrolases, hydrolytic enzymes, hydrolases, hydrolytic enzymes. Active at what? Acidic pH. They are active at acidic pH. Any pH value less than 7. Hydrolases, hydrolytic enzymes, hydrolytic enzymes. If you see these two words, automatically you should choose lysosomes. These enzymes are active at acidic pH which is close to the range of 5. Okay. Next is the second point. They are also called as suicidal bags, autophagy, autolysis. These two words are not given in NCERT, but you can learn it because sometimes when you understand the concept, if they twist and ask you, you can answer it. Okay, this is very easy to remember, so I have included this also. Auto means self, phagy means eat. Autolysis, auto means self, lysis means destruction, self-destruction. So these are also called as, lysosomes are also called as what? Suicidal bags. Write it down, students. Are you writing? Are you following with me? Or just passively watching what I'm, or just passively watching? I want you to write, students. Write it down. Just for 30 minutes we are going to do this laser focus work. Suicidal bags, suicidal bags. Lysosomes are also called as suicidal bags. Now you have to ask me the doubt, ma'am, why it is called as suicidal bag? Because of this character, that is autophagy, autophagy. Okay. Next is autolysis. Now you have a cell. Okay. These lysosomes contain acidic enzymes. Acidic enzymes. What are those enzymes called? Doctors, type the name. They are the hydrolytic enzymes or hydrolases. Now, so inside the lysosomes, you have pH which is acidic. What is the pH? Students, yeah, this is technical glitch. Now, the inside the lysosome, you have acidic pH. Outside the lysosome, what you have? Cytoplasm. Okay. What is the pH of cytoplasm, students? What is the pH of cytoplasm? Alkaline. pH is alkaline. Now inside it is acidic, outside it is alkaline. I want you to answer. Suppose if the lysosome breaks and it spills its contents over the cell, what will happen? If you know the answer, type it in the comments. Okay. So what happens? Autophagy is, there is a dead organelle which needs to be cleaned. This lysosome will go and it will round up and it will dissolve the organelle, the waste, the dead one. Okay, dead one. This is called as autophagy and clears up the dead organelle. This is called as autophagy. Autolysis means the complete lysis of the cell, complete destruction of the cell, the whole cell. Okay, all the lysosomes, it is programmed one, all the lysosomes will break open its acidic content and the whole cell will destroy itself. This is called as autolysis. Autolysis. That is why it is, lysosomes are also called as suicidal bags. They are also called as suicidal bags. Okay, if it clears up an organelle or a waste product, these lysosomes clears up an organelle or a waste product, it is called as autophagy. If it ends up in complete lysis of the cell, it is called as autolysis. Okay, now shall we write the terms, students, code words? Suicidal bags means you have to see lysosomes. They have two functions, autophagy. Autophagy. Students, are you following me up? Are you writing down? Autolysis. Autolysis is one who teaches. I am also following the same rule what I tell. Lysis and autophagy. So when you see lysosomes, you have to look for hydrolases, hydrolytic enzymes, acidic pH, suicidal bags, the organelle which is also called as suicidal bags or lysosomes, autophagy, autolysis. These words are not mentioned in NCERT, but understand, try to correlate with lysosome. Suppose if they ask in AR type, you will be able to answer. Okay. Next we will move on to macromolecular digestion. Now the question is, name the organelle which does macromolecule digestion. They can ask you like this. You will be looking for hydrolases, hydrolytic enzymes, acidic pH. I have studied everything. But suppose if they ask in the book, they didn't mention this word. NCERT, they didn't mention macromolecule digestion. But it is an indirect way to ask the four major biomolecules which is mentioned in NCERT. What are that? Lipids, proteins, carbohydrates, and DNAs and RNAs. Okay, sorry, nucleic acids. They are nucleic acids. These are enzymes, students. Yeah, these are enzymes. Okay, so nucleic acids. Here the macro biomolecules are lipids, proteins, carbohydrates, and nucleic acids. Now the organelle which helps in macromolecule digestion is lysosome. You know the answer now. You have +4 marks. What are the enzymes? Lipases, which acts on lipids. Write it, students, write it 3 times or at least 2 times. Okay, proteases which acts on lipids. Lipases which acts on lipids. Proteases which acts on proteins. Proteases acts on proteins. Next you have carbohydrases. Carbohydrases which acts on carbohydrates. Carbohydrates which acts on carbohydrates. Nucleases which acts on nucleic acids. Okay, nucleic acids. Nucleases which acts on nucleic acids. These nucleases are DNAs and RNAs. Okay, these two are enzymes which acts on the nucleic acids. Now students, are you following me? Students, are you writing up? Okay, I want you to write. Now I have a question for you. Plasma membrane. What is the, if you know the answer for this, type it in the comments. Even if you watch a replay video, what is the composition of a plasma membrane? Ma'am, I know the answer. Okay. Then, is it a fixed one? Answer, please, students, doctors. Next, which forms the backbone of the phospholipid layer? Phospholipid bilayer. Which forms the backbone of the plasma membrane? Name the phospholipid. Okay, what forms the backbone of the plasma membrane, and name the phospholipid which is involved in the bilayer. I've asked you 3 questions. Okay, if you know the answer, you type it in the comments, students. So today we are going to see the under block 2 plasma membrane, the composition of the plasma membrane. Plasma membrane means what is that? You have cell, isn't it? The membrane which surrounds the cell, which acts as a protective covering of the cell, which acts as the protective covering of the cell. Okay, so the composition which you are going to see is the composition of the surrounding layer of the cell. Inside you have cytoplasm and other organelles. What is the character of this plasma membrane? What is the character of the plasma membrane? Is it semi-permeable or selectively permeable? Answer, quickly type the answer, students. Is it semi-permeable or selectively permeable? What is the meaning of semi-permeability and what is the meaning of selective permeability? If you know the answer, type it in the comment, students. Now, the composition of plasma membrane. Okay, it is not a fixed one. You have to understand it depends upon the different types of cell. Okay, first is lipids. Lipids form the major part of the plasma membrane. Next, you have proteins. In addition to that, you have carbohydrates. Same thing again, the biomolecules. You have to study the you have to remember it as a biomolecule. Okay, one clue. Easily you can remember these are the biomolecules, right? So lipids. What is the composition of plasma membrane? Lipids, proteins and carbohydrate. Students, are you following me? Lipids, proteins and carbohydrate. Now we are going to see it as individual one. This lipid forms the bilayer of the plasma membrane. Bilayer means two layers. Two layers. Okay. One, two. Two layers. Lipids are arranged in two layers, which means this lipid part, see, we are going to study it as lipids, next proteins. Okay, carbohydrate we are not going to see. Lipids and proteins. First part, lipids. Lipids are arranged in two layers, 1 and 2. It is called as bilayer. Bilayer means bi means two. So lipids are arranged in how many layers? Bilayer. Lipids are arranged in two layers. Lipids are arranged in two layers. It is a bilayer. All these code words you have to remember. When you look at plasma membrane, look for lipids, proteins, carbohydrates. If they ask you about the composition in AR type of question, if they say like it is a fixed one, or it can be asked as a statement question. No, the composition is not fixed. It varies depending on the cell. Next, the lipids are arranged in two layers. How many layers? Two layers. What is the type of lipid here? Phospholipid. The type of lipid is a phospholipid. Under that phospholipid you have, what is the type? It is a phosphoglyceride. It is a phosphoglyceride. You have to remember this. Suppose if they add one word, phospho triglyceride. Actually, I want this to give you as a chat flash, spot the error MCQ, but I told you now. Okay. Phospholipids. What is the type of lipid, students? It is a phospholipid. Under phospholipid, what type it is? It is a phosphoglyceride. It is a phosphoglyceride. See, if they add a triglyceride, and if you choose this, you lose the marks. Okay, you have to check each and everything slowly. So it is a phosphoglyceride. Phospholipids. We'll write it one more time. A bit difficult word, so we should not forget. From here straight to the exam, we should remember everything. Okay, in this half an hour session. Phosphoglyceride. This phosphoglyceride forms the backbone of the cell membrane layer. This phosphoglyceride forms the backbone of the plasma membrane bilayer. Okay, next, the proteins are divided into peripheral. They are called as membrane, plasma membrane proteins, plasma membrane. So these membrane protein and integral protein, peripheral protein and integral protein. So before that, environment, the outside environment of the cell. Okay, now we have what we studied. We study first lipid part. So we'll draw the lipids. Okay, these are the lipids. Phospholipid, bilipid. What is the phospholipid, students? Phosphoglyceride. What is the name of phosphoglyceride? We are drawing the phosphoglyceride. Hydrophilic head. Easy, remember, this one resembles head like one, right? Head like, this knob looks like the head. If you imagine a person, how for a fun, for a small for scribbling, how do you draw a person? Like this only, right? So you name this as head. Remember this way. This is the head and this is the tail. Same way here you have head and this is the tail. So tail and tail are facing. The heads are, one part of the head is facing outside the cell, other towards the cytoplasm here. Okay, so this is hydrophilic, means water loving. This is hydrophobic. Okay, this is the hydrophobic tail part. Non-polar hydrophobic tail part. Now in this we saw about proteins next. Two types of proteins. One is the peripheral protein, which means they are attached to the membrane. Peripheral protein. Other one is the integral protein, which passes through and through of the membrane. Integral protein. I wish to explain this too. Like you have a cardboard box. For example, you have a cardboard box. Suppose if you stick the sticky notes here, it will be easy for you to peel, right? So this sticky notes is the peripheral protein. Peripheral protein. It lies on the membrane surface. It is not passing through the membrane. Okay, not going inside the cardboard box. This cardboard box is the membrane. Okay. Now integral protein means you are placing the sticky notes through as a through and through, nailing through it. Okay, suppose if you fix it with a nail, it passes through and through the cardboard box. So integral proteins are one which passes through and through of the plasma membrane, cell membrane. It gets embedded in the plasma membrane. Okay, now you got the point. Okay, peripheral means outside. It is just lying outside. That is why it is easy to extract. Peripheral membranes are easy, peripheral proteins are easy to extract when compared to integral proteins, because these are embedded through and through in the plasma membrane. Got the point, students? Now it will be easy for you to understand. So what are the two types of proteins? Membrane proteins, depending on the ease of extraction. I told you now. So peripheral proteins, they lie on the outer surface, they are easy to extract, they are called as peripheral proteins. Integral proteins, they are passing buried, they are completely buried, partially or completely buried with the plasma membrane. Integral proteins. Peripheral proteins, what are the two types of proteins? Peripheral proteins and integral proteins. Students, are you following me? Are you writing down? Are you following me? Now in this peripheral protein, see, the peripheral proteins are seen in the membrane surface. They are seen in the membrane surface. Peripheral proteins are seen in the membrane. Next you have integral proteins. They are partially, like how we nailed the sticky notes inside through and through the cardboard box. It looks like that. Okay, integral proteins are partially or totally buried in the membrane. Integral proteins are partially or totally buried in the membrane. So the ease of extraction is difficult when compared to peripheral proteins. Partially or totally buried in the membrane. Next, cell membrane is also called as, what is the other name for this? Cell membrane, also called as plasma lemma. Plasma lemma. Sometimes they won't ask you as plasma membrane, they can ask it as selectively permeable membrane. Selectively, this composition of a selectively permeable membrane. The composition of a selectively permeable membrane. These are the other words for plasma membrane. Okay. Next, what are the exceptions? I asked you know what are the exceptions to cell theory. What are the exceptions to cell theory, students? The exceptions to cell theory are viruses, viroids, and prions. These three, there are other exceptions. If you are able to find out the other exceptions, comment in the leave your answer in the comment section. Okay, what are the other exceptions? These are the Neet traps. Okay, they can ask you cell theory and they will give you the examples. Which of the following does not obey cell theory? If you have to look for virus, viroids, and prions. And we have two more types also. I will teach it in the upcoming classes. We will do the code word for that too. Next, why does viruses and viroids and prions are an exception to cell theory? Why are they an exception to cell theory? Okay, because they are considered as non-cellular or acellular. They are not at all considered as a cell because they don't have cytoplasm, no organelles. They are just genetic information completely packed particles. Just the genetic, it is completely inert. It is completely inert, that is silent. It cannot divide independently. Ask the qualification to be a cell. Okay. What are the points of cell theory? It can't divide independently. It is depending on the host cell. It does not have any cellular organs and cytoplasm. So viruses, viroids, and prions does not obey cell theory. Okay, this can be asked as an AR type of question. We can divide independently, completely dependent on the host cell. It can divide. Okay. Now shall we do a quick recap from the first. So first, what we saw, students, in block 1, type it in the comments. What are the hydrolytic enzymes? What are the names of the hydrolytic enzymes, students? Type it in the comments. How many of you have completely written all the 3 blocks at least 2 times? Because that is very, very important. I'm going to share you one small hack and then we will wind for one of the Neet tip. Very important tip, but only less people follow. This will be one among the toppers list, definitely. That is, you have bundle of books. You see everyone who recommends you see lot of videos, you buy all the books, bundle of books, then from all the coaching places, and finally you will end up with pile of books. You don't know what to study. You don't know what to study, where to start. Okay, there is nothing wrong. You can collect all the information. Okay, collect all the bundle of books, collect all the notes, PDFs, everything. But out of this, make your own notes. Make your own notes. Okay, make your own notes. So your own handwriting is very important for your last minute revision. So when you write it in your own hands, you will be easy to revise and it will be easy for you to understand in the last minute revision. Okay, understand and revision will be very easy in the last minute. So from today, make your own notes. Otherwise, file these, at least these 30-minute session. Collect all the books, collect all the PDFs. There is nothing wrong, but convert all this into your own notes, your own handwriting notes. That is why I insist on writing on your own. Okay, so follow, add now in your, take a white sheet, stick it in your study place. The first tip what you are going to follow from today is making your own notes. Not everything, the one which is tough for you, the one which you easily forget. Frame all the AR type of questions. Frame all the AR type of questions, okay? And stick it in your study place. Next, shall we start, students? The revision under block 1, lysosomes. You have to, they are vesicular membrane bound structures. You have to look for hydrolases, hydrolytic enzymes and acidic pH, okay? Next you have suicidal bags, autophagy and autolysis. Next you have macromolecule digestion, 4 major biomolecules, that is lipases, proteases, carbohydrases, nucleases are the 4 enzymes which acts upon lipids, proteins, carbohydrates and nucleic acids. These are the 4 enzymes. These are the substrates, okay? Lipids, proteins, carbohydrates and nucleic acids. Nucleases are DNAs and RNAs. They are the 2 enzymes under nucleases. Next, under block 2, we saw plasma membrane. That is, it is made up of lipids, proteins and carbohydrates. The composition is not a fixed one. It depends upon the type of cell. The lipids are arranged in 2 layers, that is bilayer. It is a phospholipid and it is a phosphoglyceride which forms the backbone of the cell membrane. Next, depending on the ease of extraction, the membrane proteins are divided into peripheral proteins and integral proteins. The peripheral proteins present on the membrane surface and the integral proteins are partially or completely buried in the membrane. Next, you have what are the other names of cell membrane? They are also called as plasma membrane, plasma lemma or selectively permeable membrane. What are the 3 exceptions and which comes under cell theory? Viruses, viroids and prions, because they don't have cytoplasm, organelles and no metabolism inside the viruses, viroids and prions, so they are considered as acellular or non-cellular. They are complete. The reason is, this is assertion, this is reason, this one is assertion, okay? Cell theory, the exceptions of cell theory, these 3. Reason is this 2, okay? They are completely inert, they can't divide independently and they depends upon the host cell for the division and metabolic activity. Okay students, junior doctors, we had a half an hour rapid fire session. Okay, leave your comments in the comment section and I am signing off now. Today we will start with another block of 3 interesting topics, subtopics from cell chapter. Have a great day students.